So now we know how to make lots of copies of your DNA. That way, every single one of your cells gets its very own copy of DNA. And we do that through the process of DNA replication. But that still doesn't tell us how DNA works. How does your DNA make you you? Because remember, it's all about proteins. Your proteins make you you. What color your hair is, whether you can digest dairy or not. It's all based on your proteins. So we need to talk about protein synthesis. How do we get from the code in the DNA to an actual functioning protein? Well, to do that, we need to meet RNA. RNA is the other nucleic acid that exists, ribonucleic acid. So it's still a nucleic acid, so it's still made out of the same building blocks, nucleotides. So let's remind ourselves what a nucleotide looks like. We have a phosphate group, which is always represented by a circle. We have a sugar ring. In this instance, it's a ribose now. And then we have our nitrogenous base, connected there by the nitrogen. But notice we have a little bit of a difference now. Before, we learned about A, T, G, and C, because that's what's in DNA. But in RNA, there are no T's. Instead, we're going to have U's for uracil. And the U is going to directly replace that T. So whereas in our pairing rules in DNA, A pairs with T, in RNA, A is going to pair with U. Okay, so it's a direct exchange there. So here are your three big differences between DNA and RNA. DNA is double-stranded. It has two strands of nucleotides hooked together by those hydrogen bonds. RNA is just one single strand of nucleotides. DNA has the sugar deoxyribose, and RNA has the sugar ribose. Okay, two different sugars. And then our biggest difference, the one they'll ask you about the most, is the bases. DNA contains T's, RNA contains U's. A lot of times the EOC likes to give you a string of letters and say, is this an example of DNA or RNA? And so you just look to see if there's T's or U's in it. There are three types of RNA that we need to talk about. You have messenger RNA, or mRNA, ribosomal RNA, or rRNA, and transfer RNA, or tRNA. Now, ribosomal RNA, that is just part of what makes up a ribosome, because remember when we learned about ribosomes, we said that they were made of nucleic acids and proteins. That's sort of their building blocks. That's how you put a ribosome together. Remember, those are put together in the nucleolus. Okay, so that's pretty much all I'm going to say about rRNA, is that it makes up the ribosomes. Now, the mRNA and tRNA we'll talk about more when we get to what their job actually is in this process. Okay, so let's do it. Let's walk through the process of how to make a protein step by step by step. Well, let's first remind ourselves of a few things. Remember that polypeptide is just another word for protein. We saw this word in our very first unit. Remember that proteins are made of amino acids. That's their building blocks. Okay, so we're going to go from DNA to RNA to protein. And remember that the nucleotides are a code. Okay, and remember that we read that code in sets of 3. 3 nucleotides equals 1 codon, and that codon is basically a code word for a specific amino acid. And as you chain the amino acids together, the protein folds up into its correct shape. So get ready and get excited, because you are all about to become international code breakers. This is your codex, or code breaker. Okay, remember that we read in sets of 3. So this is a round amino acid code. There's also a square one. I'm going to show you how to use both of them, because on the EOC, we don't know which one you will get in your questions, a round one or a square one. So I want to make sure you're prepared with both. Now with the round one, you start in the center and work your way out. So if I ask you to look up the amino acid code GCA, so you look them up in the order they're given, and you start in the center. So in the very center, and work your way out. So I said GCA. So from the center, we go to G. And then from that letter G, we go out. I said C. So here's our letter C. From there, we go out to, I said A. And then we get our answer, ALA, which is short for one of our amino acids. So ALA, that's our first amino acid. Okay? So start in the center, work your way out. Now I'm going to give you one. I would like for you to look up UUG. Hopefully you got Leu, L-E-U. Okay? So start in the center, UUG, Leu. Okay? All right, I'm going to give you one more. I would like for you to do UAG. UAG, codes for stop. Okay, stop is literally means stop, the protein's done. Okay, if you did not get those last two on your own, please pause the video and call me over so we can do it one-on-one together. You need to be able to do this. Okay. Next, let's try the square codex. With the square codex, you're going to look up the first letter you're given in the first column, and that'll tell you which row to be on. Then you look up the second letter by going across the top. Okay, that'll put you in a specific square. And then you go down the side here to do your third letter. Okay, so let's look up CAA. Okay, CAA. So we go down this column for C. We find it here. This puts us on this row. Okay, so I said CA. So I'm going to come over until I get to the A here. So that's our CA, or these four. And then I said the last letter was A. So I look over here, and this is my third A. So this is GLN. Okay, GLN. Now you try one. I would like for you to look up GAA. GAA. Hopefully you got glue. GLU right there. Okay, try one more. Why don't you look up AGC? AGC. Okay. You should have got SER, SER, right here. Okay. Again, if you didn't get this, please pause the video. Call me over to help you. Make sure that you are able to do this and do it correctly. Okay? So congratulations, you are now an international code breaker. Because there are only 20 different amino acids that exist in the world, and this code is universal. So the code for the same amino acids in every single living thing, from insects and worms and dogs and cats and fish and humans and trees and grass. It's all the exact same. That's what I think is so amazing about life, that every single living thing is made up of the four bases, A, T, G, and C. And those four bases can code for only 20 different amino acids, but it makes hundreds of thousands of different proteins and all the billions of living things you see on Earth. It's really cool. You'll also notice that using the letters A, U, G, and C, you can actually make 64 total different combinations, but there's only 20 different amino acids. You should have noticed on the code that there's a redundancy, that there are multiple different combinations that code for the same amino acid. That redundancy is a good thing. It's a fail-safe, because sometimes if a mistake is made in coding, it can still code for the same amino acid. So the protein is still made correctly. Okay, so that redundancy is a fail-safe. It's a safety measure. Now let's actually get into making a protein. Step 1 is transcription. Step 2 is editing. Step 3 is translation. And step 4 is completion. Okay, you need to make sure you know what happens in each one of these steps. Step 1 is transcription. When you transcribe something, that means you're copying it. So for example, right now I'm talking and you're writing down your notes. So you're transcribing your notes from verbal words into written words, okay? That's the same thing that's going to happen here. Now this is going to be in the nucleus of eukaryotes, and we're going to transcribe the DNA. We're going to go from DNA to mRNA. mRNA is messenger RNA. Now remember, DNA cannot leave the nucleus. It has to stay in the nucleus where it is safe and protected. But our messenger RNA that has been transcribed, it can leave the nucleus. It's a messenger. It takes the message from the nucleus out to the protein factories, the ribosomes. Okay, so that's its job, messenger. It's the messenger. It transcribes. Okay, so step 1, transcribe. Copy the DNA into mRNA. Okay. Now, this is going to look a little bit similar to what we learned before because we're going to have to untwist and unzip the DNA, and then our base pairs are going to line up. So A is going to go to U because we're making RNA and there are no T's. So A goes to U, T goes to A, G goes to C, and C goes to G, okay? This is where you need to be careful because we're going to RNA. So A is going to pair with U when we make our RNA, okay? Step 2, we need to edit the mRNA. Now we're still inside the nucleus at this point, okay? So we've transcribed, we've made mRNA. Now we need to edit it. We need to cut out the useless parts. Remember that there are stretches of DNA called introns, and introns don't code for genes. They're sort of useless DNA. So we need to cut them out and throw them out. And then the exons, the part that is the coding for the gene, get spliced together. They get hooked up together. Okay? So step 2 is editing. We just cut out the garbage. Step 3 is translation. This is the big step, okay? We're going to have codons and anticodons. Your codons are the sets of 3 nucleotides on the mRNA. We're going to have to pair them with their anticodon. That's their complementary code. The anticodons are found on the tRNA, the transfer RNA, which is floating around in the cytoplasm. Okay? So if your mRNA says AGG, okay, that's your codon, then the anticodon you're looking for on the tRNA is the opposite. Okay? So A starts with U, G with C, G with C. So your anticodon to AGG would be UCC. Okay? If you don't understand that, once again, pause the video, ask me for some help to make sure you understand. We can write this down so that you can see it. So, after we edit, the mRNA is going to leave the nucleus and go out into the cytoplasm. And it's going to find a ribosome and attach to the ribosome. Remember, the ribosome is a 2-part sphere, top and bottom. And so it's going to go right into the ribosome. And then the tRNA is going to come into the ribosome. And when the codons and the anticodons match, the tRNA releases an amino acid because that's its job, to transfer amino acids from the cytoplasm to the growing protein. This is what a tRNA looks like. I like it because it kind of looks like a wonky letter T. Make sure you recognize this. Okay. Here, the gray blob, this is the ribosome, the growing protein, the tRNAs, the mRNA. And finally, step 4 is completion. You read a special stop codon, and it releases the protein, and then the mRNA can be read again by another ribosome and make that exact same protein again. So an mRNA is not one and done. It can be used over and over again until it degrades. And you can even have 5 or 6 ribosomes on the same mRNA, making the same protein 4 or 5 times right away.